our society is quite dependent on fossil resources. And so the roles of fossil oil is actually manifold. And the first thing that you probably think about is fossil oil as an energy carrier. And from everyday life, you're aware of this, for example, in the form of transportation fuels. So for example, gasoline or jet fuels. So here it's used as an energy carrier and the oil is ultimately converted to CO2. But we should avoid accumulation of CO2 and avoid using oil. And so for cars, this can be done by making cars electric, but then for airplanes, this will pose a challenge because airplanes are more difficult to electrify. But then oil has also a second function. So oil is a carbon source. Oil contains chains of carbons which are split into smaller pieces via steam cracking. And those pieces are ethylene and propylene. And the chemical industry is using these compounds to produce most of our commodity chemicals. And important here are plastics, which are expected to still be consumed in large amounts in the future. So if we want to replace fossil oil, we also need to replace oil as a carbon source for the chemical industry. So what is a good carbon source in the future? I've already mentioned that CO2 is accumulating. So one way is to use CO2 as a carbon source, and this will be required in the future. And when you think how to use CO2 as a carbon source, you probably think about plants. So this is what plants are doing. They're using the CO2 from the air as a carbon source, and then they produce food for us, or wood, or other organic compounds. But then converting CO2 to products requires a lot of energy. And so the plants do this with the energy from sunlight. They take the energy from sunlight to help converting the CO2 molecule. So this is one way how, for example, wood can be produced, but the chemical industry wants oil. The chemical industry does not want wood, but they want a more suitable carbon source from which ethylene and propylene can be made. Further on, light is a bit difficult to access, and we need large areas to access the light. And so an energy source of the future is hydrogen, which can be made via renewable sources. And so the question then that I am asking myself, what are processes from which we can convert CO2 and hydrogen to something useful? And so to look into this, we actually go back a very long time in our timeline. So what I'm showing you here is a picture from the bottom of the ocean. This is a hydrothermal vent. And researchers believe that life has actually emerged about 4 billion years ago at this bottom of the ocean. And it's believed that these first forms of life, they have actually been eating CO2 as a carbon source. And the energy comes from hydrogen, which is formed via geological processes. And what some of these microbes are believed to do is they convert the CO2 to methane. And methane is basically natural gas. And this type of microbes or similar types of microbes are still available today. So they exist, for example, in biogas plants or also in the rumen of cows, and they make methane. And so methane is a good energy source. It can be used as energy source, but it's not suitable for the chemical industry because methane has only one carbon. And the chemical industry wants products with more than one carbon. So how can we substitute oils? We need something with more than one carbon to form this ethylene and propylene. And then an important point here is also that from ethylene you can produce jet fuels. So if you can make renewable ethylene, you can potentially make renewable jet fuels. So what I'm asking is, can we use this type of microbes to convert the CO2 efficiently, but instead of making methane, making the compounds ethane and propane that have 2 or 3 carbons, And if we can achieve that, then via existing processes, we can produce almost everything from there. And so now, in our laboratories, we work with similar microbes. We call them methanogens. These are similar to the first forms of life, and we can cultivate them in our laboratories. You can see on the left side, you can see a picture from our anaerobic chambers. So those microbes are quite easy to work with, except they don't like oxygen. For this reason, we carry out all the research in these anaerobic chambers. In the middle, you can see the picture of a bioreactor. So here we cultivate these microbes at 10-liter scale, and then we get about 100 grams of those microbes out from such a cultivation, and then we do biochemical experiments. On the bottom, you can see agar plates that are inside these anaerobic chambers. And this is a solid medium, and the microbes that we work with can actually grow on these agar plates. And this is a requirement to do metabolic engineering with these microbes. So our goal is to take this type of microbes and then make them produce something more valuable. But so to do that, we first need to better understand the microbes. I'm showing you here on this slide what the microbes are naturally doing. So they convert CO2 to methane. And this is plotted here on an energy diagram. So on the left side, you have the CO2. And on the right side, you have the methane. And then this is a multi-step reaction. So every hill is a reaction step for this process. And then, as mentioned before, this process requires a lot of hydrogen. So to produce one methane molecule, you require 4 molecules of hydrogen to help going over those hills. And now, because ultimately we want to not make methane anymore, we put a lot of emphasis in studying this step of the methane formation. Because if the microbes are not making methane anymore, we need to understand accurately what will be missing to the microbes and potentially how can we provide an alternative to keep the microbes happy. So specifically, we study the redox potential of this last step of methane formation to get accurate numbers. And then when we look a bit more on the intermediates, so each of these well is an intermediate, and you can see that whenever a hydrogen molecule comes in, the carbon will have 1 hydrogen more. So from the left side is the CO2 activation, which is the highest hill, and then the carbon will be converted to a formyl group that is bound to a nitrogen atom. And now these intermediates are not free intermediates, but they're bound to a cofactor. And in methanogens, they're bound to a cofactor called H4MPT, which is a derivative of folate that you might be familiar with from your own body, which is also termed vitamin B9. So this cofactor helps to promote these 1-carbon reactions. And then this H4MPT variant has evolved for efficient CO2 conversion. But now these intermediates are only known with 1 carbon, but ultimately we want to produce compounds with more than 1 carbon. And so recently we could isolate a cofactor that contains a C2 unit. So here you see this H4MPT with an ethyl group bound to it. And so this research shows that this cofactor can carry multi-carbon compounds. And we are now studying enzymes that can process such compounds, and ultimately we want this ethyl group to end up as ethane as a product. And so next, we study reactions to form carbon-carbon bonds, because carbon dioxide has only 1 carbon, so we need new tools to assemble carbon-carbon bonds. And to do this, we get inspiration from nature. In nature are actually microbes that do the opposite of what I want to do. So they're microbes that convert ethane or propane to CO2. But then, because these metabolisms are reversible, we want to learn how nature is breaking carbon-carbon bonds and then using the same reactions to form carbon-carbon bonds. And also these microbes contain interesting enzymes that can transfer C2 and C3 units. But then how do we convince the microbe to do something different? So what the microbe is doing is encoded in the DNA. And so if we want that the microbe is doing something different, we need to change the DNA of the microbe. And so for this, we have developed a so-called CRISPR toolbox. This is a modern tool in synthetic biology to more precisely and more rapidly change the genome of the microbe. And now, the purpose of Aalto-University is to shape a sustainable future. And so this means we are not only doing fundamental research, but we want to translate the research into impact. And here I have summarized the research areas that we are doing. So we want to use the metabolism of methanogens. When we understand it correctly, we can start amending it. We have cofactors that can carry multi-carbon units. We take inspiration from nature, how to form carbon-carbon bonds. And then with the genetic toolbox, we can actually start designing our own microbes with the aim to have microbes that convert CO2 to ethane or propane. So to summarize, the research that we are doing in my research group is focusing on the native capabilities of CO2 conversion found in these microbes called methanogens. And then we apply tools in synthetic biology that the microbe can produce more valuable compounds to feed into these existing value chains. So the overall aim is to help our society and the chemical industry to become independent of fossil oil. Thank you very much.